This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From Microbe TV, this is TWiM, This Week in Microbiology, episode 139, recorded on November 8th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, how are you, Vincent?
I'm very well. And yourself?
Good, good. Enjoying the good weather here.
It's beautiful. It's nice fall weather, but there it never changes, right?
Mm.
The leaves don't fall off the trees, right?
Right.
I have to pick up leaves every weekend, but I'm sure you remember from your days in Boston.
Absolutely.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
Where the leaves are also falling, right?
They are, but we had a beautiful 60-degree sunny day for President Obama's visit yesterday.
Oh, did you go?
I didn't. I was teaching class, but I did excuse any student, but I said they still had to do the assignment and send me a selfie.
Nice.
From the rally.
Oh, that's great. I would love to go. I would love to go even when he's former president. And I would love to see Michelle Obama in person.
Yeah. Yeah. She's amazing.
She's such a great speaker. Well, you know, the rumor is he may be coming to Columbia. So maybe I'll get to see him.
I was hoping he would be coming to TWiM.
Well, you know, if he's at Columbia, maybe I could talk him into it.
Yeah.
We could do it. That would be fun. I could have him here with a Secret Service detail, right? Well,
that would be interesting.
Also joining us, as you can hear already, from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
No travel for you. You're traveling tomorrow, aren't you, Michael?
I'm traveling to the great state of Texas to celebrate the 75th anniversary of the Texas branch of the American Society for Microbiology, and also witness a milestone event at UT Southwestern in Dallas.
Oh, that's nice. A lot of good microbiology history there, right?
Oh, yes.
Yeah, enjoy that. Those are always very nice. There was recently one at Merck not too long ago. And there's a lot of microbiology history there as well.
And just in case our listeners aren't familiar, the Milestones in Microbiology designation is awarded by ASM to recognize the contributions of an institution to microbiology.
And we did one out at Ann Arbor last year,
right.
Was it, right?
Yeah, but I think there are only, a dozen or maybe 14
not too many.
Sites, so it's a very elite honor.
That's right. And you get to have a little ceremony, and then you get a plaque to put on the wall, which explains the contribution and so forth. So I've been to a few of them. I was one at Cold Spring Harbor and the one in Ann Arbor. And you went to Wisconsin, right?
To
Michelle?
Madison,
Yeah.
Yep, University of Wisconsin.
Wonderful. All right. Today we have a couple of interesting papers for you. Here in the United States, it's election day, so we're all on pins and needles. But we're going to distract ourselves with microbiology, or shall I say the microbial sciences, which are just awesome. And as Michelle said, we have a little control over those. Maybe.
Well, that's our delusion. The microbes are actually controlling us.
They are controlling us. But we work together, really, don't we?
It's a partnership.
Give them a place to live. They give us things in return. So I have a snippet which is actually related to this whole topic of mutualism. And it is a paper that is not yet published, but by the time this episode is released, it shall be. It shall be published in M-Sphere, the open access ASM journal. And it's called Microbial Community Patterns Associated with Automated Teller Machine Keypads in New York City. And the authors are Holly Bick, Julia Maritz, Albert Long, Hak-Dong Shin, Maria Gloria Dominguez Bello, and Jane Carlton. They are from the Department of Biology at New York University and the microbiome program at New York University School of Medicine right here in New York City. And this is kind of a cute study where they're looking at the urban microbiome. This is part of the field of urban microbial ecology to try and describe the biodiversity and the biogeography of prokaryotes and eukaryotic microbes. And in this particular paper, they're looking at ATM machines in New York City. And apparently this is an unexplored habitat. Here is a factoid.
There are a few left, are there?
Yes. Here's a factoid, which is in the paper. In New York City, the surface area of urban surfaces far exceeds the geographic area of the island. So the built environment, ATM surfaces, subways, it's more than the area of the island. And there have been quite a few other studies done so far looking at the microbiome in the subway, mass transit systems, in the soil of the city, in plumbing and ventilation systems, in buildings. But apparently the ATM has not been done before, so that's what they're doing in this paper. I know a guy in Brooklyn who gets soil in New York City and looks for parasites that dogs might put there. And people do all sorts of things here in the city. So ATMs are a good candidate because they're obviously high traffic devices for the human touch, right? People are hitting them all day long and lots of different people. So what they did, they collected 66 samples from ATM keypads across New York City. Most of them were inside, indoor ATMs, but a few were outside. And they looked in 8 neighborhoods in Manhattan, Queens, and Brooklyn. For those of you who know the city, they looked in Midtown Manhattan, they looked in Kips Bay, and they looked in Chinatown. They looked in Central Harlem and Marble Hill Inwood, which is not too far from where I am right at this moment. Those are all in Manhattan. Then they looked in West Brighton, which is part of Brooklyn. They looked at South Ozone Park and Flushing, which are both in Queens.
So, home of the Mets.
Home of the Mets.
Flushing,
right.
Home of the Mets. That's right. Who once won a World Series, I think.
Well,
yeah.
But we can talk about that later.
And I think the coolest part of this, let me digress on this election day for just a moment. They looked at these 8 different neighborhoods, and they have a wonderful figure showing you the diversity that is here in New York City. They have, Manhattan areas, they have Queens, they have Brooklyn, and the percent foreign-born of the inhabitants varies from 22 to 70%. In Flushing, 70% of the inhabitants are foreign-born, mostly Asian, but the dominant ancestry is Italian. The Italians are also the dominant ancestry in Kips Bay, which is the Murray Hill section of Brooklyn, which is predominantly white. But so many other areas, Marble Hill Inwood here in Manhattan, 75% Hispanic Latino, 47% foreign-born. The dominant ancestry is Irish. Take a look at this table. Every area is different. Irish, West Indian, Italian, Russian, Latin America, Europe, Asia. This is...
Let me ask you, is that the whole neighborhood or a selected subset of the neighborhood? I wasn't clear about that.
So these neighborhoods are made up of these numbers, the whole neighborhood, not all of Queens, but just say South Ozone Park or Flushing. Uh-huh. It's, they do censi.
Yeah. But they're selected in a way. I mean, this is not a random distribution.
It's a neighborhood tabulation area, which is a census term that a lot of the demographers use for determining what's actually going on in a particular area. It's used a lot in health outcome studies where they're looking for blood pressure and they're looking for diabetes incidence and all sorts of other things. And so they're trying to make sense whether or not there's a founder effect, and they use these sorts of tables for cancer. That's why South Carolina is known as the buckle of the stroke belt, because of where we sit and the incidence of stroke and cardiovascular disease. If they looked at a belt across the United States, we are the buckle of the stroke belt, or so I hear each time I go and attend a seminar on stroke. So this demographic
speaking
table
of open access, the data are available in the figure legend. They give you the URL, so you can go and look at this data sets also.
That's right, New York City Open Data. Anyway, I wanted to point out this incredible diversity in New York City. I'm proud because my dad came from Italy many years ago, and now the people continue to come into the U.S. The U.S. is made of immigrants, obviously, and today I'd like to celebrate that.
And it's why you have such awesome food in New York City.
We have great food, and
great
we have
groceries.
great mines. This, mines come in from elsewhere, and it builds this country. So this is wonderful.
Pollinates.
I completely embrace this. Anyway, as I said, 8 neighborhoods in Manhattan, 66 samples. What they did, they took cotton swabs that were moistened with a saline TWiM solution, and then they scrubbed all the keys with two swabs for 10 seconds each, and then they put them in a tube and took them back to the lab. They did control swabs where they just had them moistened. They held them up in the air for 10 seconds, to collect anything.
And meanwhile, there's a line of people wanting to deposit a check behind them, right?
Yeah, these people are like, hey, come on. What are you doing?
Another nut in New York City.
Hey, I resemble that remark.
Resemble that remark.
Well, we sampled in Grand Central Station, so we got the looks too.
10 seconds. You sampled for what? Copper?
Yeah, we were assessing the bioburden on the Grand Central Station's beautiful copper rails in the Great Hall.
Oh,
cool. All right, so then they take these swabs and they bring them back to the lab. They extract RNA, DNA, and they sequence 16S and 18S ribosomal RNA sequences. And for those of you interested, they use an Illumina MiSeq platform. Now, remember, this is microbial DNA. This is not microbes, right? And it's very different.
That's right.
It's just DNA. We don't know if there's any viable microbes. There must be some, but it's hard to know. There'd be another study. They actually say at the end.
I think that's a key point. I'm sorry to interject.
Yeah, it's okay. Sure.
We can talk about this later. I don't know that these studies tell you anything because they don't tell you what the consequence is. Supposedly, if you find bacteria in a certain place, it's because you worry that they may be transmitted. Well, if they're dead, they're not going to be transmitted. So why do this study this way? Why not include right away, as part and parcel of the study, the determination of viability? I don't understand.
I think that would be good. In fact, of course, not everything's going to be viable because not everything is culturable, right?
Right.
Which is part of the problem.
I suppose that most of these bugs are dead, so it's a big fat so what?
Well, I guess you can say that somehow this microbial DNA got here, and where did it come from? But we don't know how long it was there, right? So that could be an issue as well. So yeah, I think this paper, when it comes out, is going to get a lot of press, I think.
Oh, yes.
And it should.
Yes.
I mean, I honestly don't understand the logic of doing this kind of study in isolation without including viability studies. I don't get it.
That gets much more complex and much more expensive. So they may have done this as a first pass, and then depending on what they saw, design the other study.
Right. You can do a million things like with this because it's easy, it's relatively cheap, but it doesn't make sense.
If they have these swabs that they
brought back
and they properly preserve them,
doesn't mean you have to do it.
If they properly preserve the swabs, perhaps they could go back to them and try and culture. But I suspect they used it all for DNA extraction.
They most likely extracted. Yeah. We struggled with this a lot, and I've been partially brainwashed by our colleague Elio here. And I always ask the question first: who's there from a viability standpoint? And what we do is we freeze a portion of the culture liquor that we were covering, the PBS TWiM and lecithin mixture, and then we add, you may screw things up by adding glycerol, so we always freeze half with glycerol and half without in order so that we can extract DNA or RNA cleanly if we're looking for viruses. And we haven't gone back to do the genetics, but we've done the viability studies. And it's always a toss of a coin as to what you do first. This will give you a tremendous amount of information, as Vincent's going to tell us about. But at the same time,
even your approach, Michael, presumes that you know what media to use.
Yeah, that's right.
Oh, we use a shotgun approach, Michele. We use a lot of different flavors.
Yeah, that's a good point because not just that, but the primers you use to amplify the ribosomal RNA genes, the sequencing platform that you use, they point out in the paper that can all affect what your outcome is. So it's not definitive. You can just look at it as a survey. I don't know how costly these are to do, but maybe they're easy enough to do that this is a first pass. Anyway, those are the caveats now that we've set them out ahead of time. Let's see what they found. First of all, their controls revealed a lot of microbes in the air, but also in the cotton swabs. They're not sterile. They have DNA. Well, they're sterile, but they have microbial DNA in them, right? Even in the lab reagents, there's microbial DNA. So,
that's how sensitive this method is, huh?
Right. And they have to subtract that from all the data sets, right? So they immediately say, we're subtracting all of these sequences from the data set before they analyze it. So what did they find? So they have two different data sets, the 16S and the 18S ribosomal. Of course, 16S is for prokaryotic, 18S for eukaryotic. In the 16S, the most abundant phyla were Actinobacteria, Bacteroides, Firmicutes, Proteobacteria. And these basically are representative of the human skin. At the class level, Actinobacteria, Bacilli, Clostridia, alpha Proteobacteria, these are widespread and metabolically diverse group of environmental bacteria, and they have been found on urban transit system surfaces before, and gamma Proteobacteria. Now, in the archaea, they only find 9 what they call operational taxonomic units, OTUs, which we've talked about before in terms of using sequence to identify groups of microorganisms. And they're very low abundance, and they say this might be a problem with the primer sets that we're using, so something that we have to go back. The 18S data set, again, eukaryotes, fungal OTUs were the largest taxonomic proportion of those, and then next were metazoa, and then unassigned OTUs, sequences they have no idea what they are. They don't come up in any database. There are a lot of them, and we don't know what they are. They also find protist lineages, amoebozoa, and alveolata, which are free-living ciliates. They're often found in fresh water and soil, and rhizaria. These, however, are pretty low abundance. A couple of details here which are interesting. Aspergillus niger was enriched in the central Harlem South samples. This is a ubiquitous fungus. Of course, it's found outside. It's also found indoors in the air. It's used in industrial processes, and it's a cause of black mold disease in many fruit and vegetable crops. So many possibilities about where that came from. Another fungus, Occultifer externus, was also enriched in the central Harlem South samples. This is a recently described novel species of fungus originally isolated from plant litter in Portugal. Xeromyces bisporus is a foodborne mold found mainly in the white neighborhoods that they sampled here. Often it was originally found in licorice, and has been associated with spoilage of high sugar foods like cakes and confectionaries. And the neighborhoods where they found this have a lot of bake shops, so people probably go and they buy a cake or something sweet, and then they go to the ATM machine, and it's on their hands.
Sticky fingers.
Sticky fingers. In West Brighton, which is right near the water in Brooklyn, one of the amoebozoa was over 1% of the samples. And in Midtown, an entamoeba species. And if those of you know some of the parasitic infections of the gut, entamoeba histolytica, this was there in Midtown. It's an intestinal tract isolate, of course. One sample had over 10% Silicophilosia protist, which was West Brighton. This is an amoebal member of the Rhizaria group from soil ecosystems. They also recovered various trichomonads, both free-living and animal-associated, so some suggestion of passage between animals and humans. They also found toxoplasma, over 3% of the sequences at 1 ATM in West Brighton. Toxoplasma, of course, is a protozoan parasite. I asked my colleague Dixon Despommier. He said, that's because all the Russians there like to eat raw meat.
And cats. And have cats.
And they have cats, and that's why you have it so high, maybe in West Brighton. Who knows? Okay, the metazoan taxa, multicellular animals, bony fish and mollusks in samples from Asian neighborhoods, and in black neighborhoods, chickens, sequences from chickens. The authors call this a molecular echo of the foods that predominate in those areas. Of course, they don't know the source of these DNAs on the ATM, but they say, well, these people are eating these particular foods, and then they go to the ATM machine, and the DNA is left over on their fingers, or a little bit of food. Sticky fingers. They say that meals contribute very much to the urban microbiome. In laundromats and other stores, they had some ATMs in those. They found the highest number of taxa on those ATMs, with a lot of lactobacilli, but others as well. And I guess, I don't know, when people are handling their clothes and putting them in.
It's the underwear effect. The lactobacilli are associated with those garments that are close to our skin, where the microbes are. It's the underwear effect.
That's a good point. And you're handling them?
Skin, dead skin scales.
And dead skin. But it's really, most people are good about washing their underwear.
Right.
And you rarely staff that out.
All right. So all of that's sort of an overview. But the surprising part, or maybe not so surprising, there's no clear pattern of communities across the ATM keypads in New York City. It's not based on geography, the location of the ATM, the local population, the time of sampling. And they try and explain or think about this a little bit in their discussion. One of their ideas is that ATMs are high-use locations, and you get a lot of people not just in your neighborhood, but maybe travelers from outside the neighborhood. And they say this could homogenize the microbial communities on the ATM. So you have transient users. Transient users will eliminate the community signatures of the ATM machines. Also, some of these may be cleaned or disinfected. I've never seen anyone cleaning an ATM. I've never seen that, but that could have an effect as well. But they also suggest that maybe, the primers, the sequencing platforms, and, to get the identity of the origin of these DNAs, they have to go to databases, and the databases, the data they are acquired in different ways. So that may contribute as well. So what's the source of these microbial DNAs? Most are from unknown sources. They can't identify them. They don't hit anything in the database. Less than 25% of the sequences were from known sources, and these include household surfaces like TVs, restrooms, kitchens, pillows, and then humans, hands, palms, nose, ear, intestines. But they emphasize that the human sources are not a primary source, very low proportion of all the sequences. And again, they think because these are high traffic locations, the results reflect kind of the average community pooled from different sources. And finally, in their discussion, they get back to the discussion we had earlier. They can't distinguish between active and non-active microbes. They don't know how long they've been there. And so they suggest maybe they should start looking at culturable microbes, or at least metabolic activity and so forth, to confirm some of this. So it's kind of interesting to see what's present. I was particularly interested in the food, and I think the main take-home message is that people need to wash their hands a little better.
Well, if it's just fragments of DNA, it's probably not doing any harm.
Yeah, I suppose. But they got food on their hands. They probably should wash them, right? Or maybe it's just so sensitive that you've washed your hands, even you have a little bit of food left, right?
Right.
It could be.
But I guess the good news in this study is the no news. It's not like they found MRSA
that's true.
all over New York or something else dreadful.
Yeah, they found only a couple of potential pathogens, and they say these have to be verified because the sequencing might be getting something close to them, but not exactly the pathogen. So
I don't doubt that studies like this should be published, but I think there should be a specific journal which is a repository of just raw data like this. Mm-hmm. It's probably worth having this at hand, but there's no message here other than a very negative one.
Yeah. To be fair, they didn't know that at the start. They did this survey and found what they found. And at least one thing I appreciated, the discussion, they were very clear on what the limitations were and what the technical biases introduced by their methods were. So they gave us a very good discussion of this type of approach.
Yeah. I think they were also clear on the limitations.
Very honest.
Well.
Yeah.
Elio's right in the sense that it would be nice if we had a place to put all of these data associated with for lack of a better term, the built environment. So, and we're facing this same issue with healthcare as we begin to collect all data on people now that electronic records are becoming more common in healthcare. We're collecting large volumes of data for which we have no way of really analyzing it with some of the new analytic tools that companies like Google are really pioneering, just trying to understand how Americans and the rest of the world buy things. Just imagine if we were able to evaluate that from a perspective of risk. I think the question here is, do ATMs represent a risk? And right now the answer is no. From the data that they provided to us, ATMs appear to be perfectly safe. They're effectively collecting natural skin flora, and they're reporting what we're eating, and they're reflecting on what's in the neighborhood and maybe depositing from air currents as the ATM door opens or closes. But there's really no way to assess whether or not an ATM represents a risk. And I think that was the meta question that they were trying to get at. And as we begin to use sequencing and it becomes much more inexpensive to develop these data, I think a common repository would be good. And then the real work of data mining will come into play to ask the questions about risk and asking Elio's fundamental question, what does all this mean?
It's also would be a tremendous waste of resources if we didn't publish our data that didn't come to a striking conclusion, because then others can read it and say, I guess if I'm interested in that question, I'm going to need to design the study in a different way.
Sure.
So we don't have lots of people doing the same thing and coming up with inconclusive or uninteresting results.
And I think the common sense conclusion here is that if you go out in public and touch things, you should wash your hands before touching your own mucous membranes.
Absolutely.
But that's
True.
always been true before we had
or
these
the mucous membranes of your loved ones.
Yes, of course. Thank you, Michele. Let me tell you about the sponsor of this episode, CuriosityStream, the world's first ad-free nonfiction streaming service founded by John Hendricks of Discovery Channel, so you know you're going to get real science shows. You can watch these movies or episodes on the web or on any of a number of devices that interface the web with your TV, like Apple TV or the Roku device. You can find it in 196 countries, and they have a wide variety of science, technology, nature, history, interviews, lectures. A cool one that they have, which I've just discovered, Stephen Hawking's favorite places. Stephen Hawking takes you on a CGI spaceship across the universe, and he makes stops at some of his favorite destinations. That's cool. Digits, a 3-part series hosted by Derek Mueller, the creator of YouTube's science channel Veritasium, which includes interviews with Edward Snowden and Vint Cerf, deep time history, documentary on the 14 billion year history of the Earth, and many, many more. Lots of good science there. They also have a very big, super high definition 4K library, over 50 hours of content. They have monthly and annual plans available starting at $2.99 a month, less than a cup of coffee. Check out curiositystream.com/microbe. Use the promo code microbe at sign up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiM. Let us now turn to Michael for our paper today.
All right, so I was going to save this paper for when Michigan was playing The Ohio State University, but thought it was ready to go. And this.
Do you mean that Michigan team that's number 2 in the country and 9-0? Is that
I
the
do
Michigan
indeed. And so I was going to lend some support to Ohio State since they needed something to boost their morale since the thumping they are going to get when they play Michigan. So this paper is entitled Microbial Metabolism in a 2.5 kilometer deep ecosystem created by hydraulic fracturing in shales. This paper is by Daly, Borton, Wilkins, Hoyt, Counts, Wolf, Welch, Marcus, Trexler, McRae, Krasicki, Cole, Moser, and Wrighton. And they are from The Ohio State University, where they're in the Department of Microbiology and the School of Earth Science, as well as the Pacific Northwest Laboratories in the Department of Civil and Environmental and Geodetic Engineering, also at The Ohio State University. So
and some from Maine.
Oh, excuse me. I forgot one. I apologize. And also from the University of Maine and Orono. And
Orono.
Okay. So
Sorry.
that's okay. You feel free to correct my pronunciation as you wish. I apologize if I ever botch anyone's name. It's not intentional. So we've all benefited from the lower gasoline and natural gas prices as reflected in our respective bills from the gas pump and our electric companies, though my fair state has a pro-greenhouse free energy. And so I've actually seen a 37% increase in my utility bills because they're building nuclear reactors. But that's another discussion for another time. But attention has been paid to the economic benefits and environmental impacts of hydraulic fracking of our Earth's crust in order to obtain hydrocarbon. Yet the biochemical changes induced in the deep subsurface are poorly understood, and evidence of this occurred on Sunday, this past week on November 6, 2016, where there was a 5.0 magnitude earthquake that shook central Oklahoma. And this is a big earthquake because it was felt as far away as Dallas and Kansas City and Little Rock, Arkansas. And again, this took place in central Oklahoma. And the United States Geological Service has recorded over 1,000 earthquakes of magnitude 3 or greater in this region of Oklahoma, nearly 3 times as many as the 318 former earthquakes that occurred in 2009. So you think about it, earthquakes are one symptom of what the consequence of a fracking is in order to release the oils and gases that we're all taking advantage of in our utility bills and lower gas prices. But as microbiologists, we need to be asking, what's the unintended consequence of fracking going on with the microbes as we're injecting microbes likely into this region where we're effectively pumping in drilling fluids that have nutrients in. So we're actually feeding the microbes that may be in the deep subsurface, as well as we're introducing microbes because these fracking fluids are not sterile. We're actually injecting nutrients. And so the story in brief is an absolutely fascinating lesson in microbiology and or systems thinking if you really set your mind and begin to think about what's going on. So the hydraulic fracking input fluids from both the Marcellus and Utica shales, and I put in the show notes where the Marcellus and Utica shales are located, and the Marcellus shales are effectively located in most of Pennsylvania. And the Utica shales are located in upstate New York, and there's a nice little map in the show notes to give you an idea of the range of these shale formations. And in fact, the Marcellus shale represents about 60% of the total landmass of the Commonwealth of Pennsylvania. And shale gas accounts for 1/3 of the natural gas energy resources worldwide. And in fact, in today's current events, you saw that there was a deal cut with the state of Iran where they're actually going to begin to exploration for gas using some of this technology over in that area of the world as well. So back to the microbiology story, the recovery of the gases depended upon this hydraulic fracturing technology where you had the high pressure injection of water and the chemical additives. And the chemical additives in this cutting solvent are monomethylamine, ethylene glycol, methanol, choline, dimethylamine, and sucrose. And you don't have to be all that familiar with the metabolic processes of microbiology to know that sucrose is a great carbon and energy source, as are many of the other materials that.
Briefly, why do they add all this stuff?
They add the stuff because it facilitates the drilling process, and it also facilitates the recovery of these sticky materials from the rock. And that's the extent of my geology knowledge. I'm sure we'll get notes from the geochemists in our audience that will school me on the cutting fluid cocktail and the hydraulic fracturing fluids and why they're useful. But from my selfish point of view, they were effectively answering the question, what do you need to grow bacteria? And you need a source of carbon, you need a source of nitrogen. And so in methylamine, you have both. And so, it's really all about carbon. You already got the sulfur present because many of the hydrocarbons are rich in sulfur. And so you're really doing microbiology 101 with the ability to grow these things. So in brief, what they were able to show is that they did some preliminary data that have been previously published and using single gene investigations, and they found halotolerant or salt-loving microbial communities were actually enriched subsequent to hydraulic fracturing. And what they did here is they reconstructed, and again, we're going back to the molecular reconstruction of genomes, and they're actually characterizing the genomes of the Marcellus and Utica shales, and they reconstruct these genomes, effectively taking samples over a time course, asking how does the community change with time, and that's effectively the story of their paper. And then they ask a couple of other questions about how it's going on, what's going on, to yield the final take-home message that hydraulic fracturing provides organismal and chemical inputs for the colonization and then persistence in the deep terrestrial surface. So not only are fracking is not only causing earthquakes, but it's also giving Mother Earth an infection.
Michael, this is 2.5 kilometers down that they're getting these samples?
Yep.
Now, do they have controls from sites? I guess not, because there's no pipe going down there, right?
No.
So they can't get a control and say, what's there without us adding stuff, right?
Yeah. There's no way to get down there without drilling. And you can't drill in a dry sense, so to speak.
Yeah, of course.
But they do go on to say that the species that become dominant were introduced from the surface.
Correct. And I was going to get to that when I get to the
right.
detail.
Sorry.
This was preamble. And so what I want to have the audience do is when you go and pull this paper, and this paper is in Nature Microbiology, and it's an open access paper, I believe, because it has the word open, at least on the manuscript I downloaded. So I assume it's an open access paper, so you should have the ability to get it. I encourage you to look at their fourth figure before you even read the paper. And the reason is figure 4 gives you the overview of the lesson in systems thinking that these folks use to approach this problem. And what this figure describes is the interconnected metabolism or metabolic profile that is catalyzed by persisting microbes in these hydraulically fractured shales. And so you're fracturing the shale to release the gas because they're after natural gas in the Marcellus shale. And the consequence of this is what they take you through. And step 1 is they inject this nutrient material in, and that begins to break everything up. And then what happens is you see the enrichment of these salt-loving bacteria that then go through this metabolic opera where they do all sorts of remarkable things. And as you look at this figure, it'll set you up to take you through the basic premise of their paper, where they ask the question, who's there over time? Then they ask the question, how are they dealing with the salt and what's actually going on? Then they ask, are the microbes impacting energy extraction? That is, are they sampling the goods? Are the microbes eating the hydrocarbons? And then they introduced a twist in their paper, namely, they introduced viruses and the viral predation of the microbes that are actually a consequence of the injection of these chemicals. The viruses are actually preying on the microbes that they're putting in, and they have evidence that there's active viral predation in the deep subsurface. And then they asked the question that our former paper asked about whether or not there's differences between shales extracted from the Utica region versus the Marcellus region. So let's just take you through the data. Step one, asking the question, who's there in the shale genome? That is to say, they go in, they have an input fluid, and they're asking the question, who's there at input? Asking the question about, what's in the cocktail? They then extract the fluids at 7 and 13 days, which they consider early, and then again at day 82 and day 328, which they consider to be late time points. And as you look at their first figure, they have this cute little growth curve snuck in. And what the input fluid is injecting into this well is about 50,000 colony forming units per mil of fractate liquor that they're injecting in. So they're putting about 50,000 bacteria in to the shale when they begin to frack it. And then at day 7, what you see is a little bump, or what we would, as a microbiologist, call a little lag period. By the time you get through that first week, first 2 weeks, if you will, at day 13, you see a little bit of a rise. And by day 82, we're beginning to see logarithmic growth. By the day of 328, the concentration of bacteria per milliliter of the hydraulic fracking fluid that they recover is well over 1 million CFU per mil. And so
let me just emphasize that is the total population. So they don't yet know with that figure whether there are some populations that are declining and others taking over, et cetera. But you're going to get to that, right?
I'm going to get to that. And that's the beauty of, and in their figure, they use different colors to highlight different bacteria and different bacterial genera. Now, remember, they did this not by culturing, but they did this purely by DNA sequencing. So they're taking a very complicated mixture of stuff out of Mother Earth, and they're extracting DNA and they're assembling genomes, asking the question, who's there? And what they learned is that there is indeed a succession of bacteria that do indeed change with time. And as you begin to go through the data, you begin to see how they change. And we see an evolution of the community structure. It's not evolving in the sense that it's making mutations. It's evolving in who's there in dominant numbers. And that is quite evident from their color pictures or their color graphs that they have for you to see what's going on from the perspective of the genome bin relevant in terms of the relative abundance. And they did this in replicates. They did this 3 times, and consistent with their early taxonomic study, they found that 6 halotolerant bacteria and archaea became enriched at the later time points at day 82 and 328. And they were both halo and thermal tolerant. So they were resistant to both salt and heat. And the environmental sequences closely related to the 16S ribosomal RNA genes that they recovered were similar to those that have been published for other hydrocarbon reservoirs or other hypersaline environments. And then they are proposing a new bacterium that they've identified from this, and they've appropriately named it Canadensis Frackibacter.
I love it. Frackibacter. So that means, Michael, it's unique to the shale, right?
Correct.
It's the first place it's been discovered.
Yeah.
It's the first place it's been discovered. They did not detect the lower abundance members of methanogens and Frackibacteria in the input fluids. And they rightly point out it's because it may have been in the input fluids, but it may have been below their limit of detection. And so what's happening is, by virtue of the fact that they're putting this stuff in and they're incubating it in a present of high temperature and high pressure, greater than 25 million pascals and higher salinity, is that you're effectively fracking generates this habitat where low abundance microbes injected into the deep subsurface can bloom and persist despite biocide addition and these elevated temperatures and hypersalinities. The second aspect to their paper is how is the community resisting this hypersaline environment. And halite dissolution from shale matrix drives large salinity increases in the produced fluids, or what they recover. So they comment about how the organisms must have adaptations for tolerating a broad salinity range. And this is where the genetics or the sequencing infers metabolism. And they begin to talk about the assembly of these genomes, and they denote that there are genes present that will enable it to resist these things. But recognizing that we'd likely talk about this paper, and Elio would say, so what? They also
No.
But I'm getting to the fact that they quantified the metabolites by NMR and chloride in the fluids from this Marcellus shale. And what they see is at time zero, they have the input fluids going in, and they have both the Utica and Marcellus shales in their second figure. What they noted is that as consistent as a response to salinity, they make this product glycine beta-ine, which is a osmoprotectant to the microorganism. So as the chloride goes up, so does this osmoprotected component. And so this GB, and that's the abbreviation that they use, or this glycine beta-ine, was present in the fluids, and it was below detection in the input fluid and early Marcellus fluid, but then it reached a maximum concentration at 82 days, and it was maintained at that high level through day 328. So it's indicating that the community is making enough of this GB, which is an osmoprotectant. And then here's where it gets really neat, is that this GB synthesis is encoded by a methanohalophilus genome by the glycine pathway, and in one of the other predominant organisms they recover, Halomonidaceae and the Marinobacter genomes, and that's encoded off of a pathine from choline, which of course is one of the additions that they put into their input fluid. Is there's a large
component of the
component
fracking fluid.
of it, and so it's effectively serving, if you will, as an enrichment. And this choline is common additive in fracking fluids, and it gets consumed by day 13 when Marinobacter becomes abundant. So the take-home point here is inputting a precursor is fueling this microbial bloom through this production of a haloprotectant. And so now here's where do no harm needs to be addressed. The microbial synthesized GB is shed into the fluids as an osmoprotectant that then can be microbial degraded by both the recovered obligate fermenters. And the obligate fermenters that they have are these halon aerobium and the Frackibacter. And I thought this was very interesting, is that this glycine beta-ine fermentation is using microbial produced metabolites rather than a dependency on the input fluid chemicals. So again, this is the exercise in community microbiology showing how the community is sustaining life in these shales long after fracking has done its job and, effectively busted up the gases so they're diffusing up, going up to the wellhead, which is sustaining the bloom. But the danger to the oil company is as you continue microbial activity, you could effectively sour the well. Just as when we have milk that sits in our refrigerator too long, the milk can sour because the milk is not sterile. So this fracking environment is not sterile, and the gases can actually sour and begin to cause all sorts of unintended consequences.
And chemistries.
And chemistries, absolutely. So part 3 of their story takes us then into the metabolic pathways that then influence the economics or impact the energy extraction story. So in addition to the choline that supports the bloom of these salt-tolerant microbes associated with the hydraulic fracking fluids that contain high concentrations of the organic substrates such as sucrose, and the sucrose that they're adding is actually pretty high. It's 0.3 millimolar. And the ethylene glycol, or the antifreeze that we all put in our car, is actually going in at 3.6 millimolar. And the community will respire sucrose and consume ethylene glycol. So you can see then how all of this will impact the subsurface by simply looking carefully at their second figure. And when you look at their second figure, it gives you a story of what's going on. You see the osmoprotectant, the GB, matching or mirroring as the concentration of chloride goes up, which is an indicator of salt. So does the osmoprotectant, GB. And then you begin to look at the consumption of the materials that they're putting in, like ethylene glycol. It literally craters very, very quickly, as does the methanol. And then they look at these other things that are going up, and that's indicative of other microbial processes going on. Namely, acetate is actually going up, and then you begin to see the acetate begun to be consumed, and it's likely going into methanogenesis or some other component of the integrated community response to all of these input chemicals that are actually being present. And so they then begin to ask the question, are the microbes sampling the goods? In other words, are they degrading the hydrocarbons, which could impact on the ability of the well to remain stable to get as much of the energy out for human use as opposed to microbial use? So having the shale produce fluids, they look for the degradation pathways associated with eating the hydrocarbons, namely BTEX. And that stands for benzene, toluene, ethylbenzene, xylene, and naphthalene. And that's the N in the BTEX-N. And in all the fluid samples, they found that the bakers were actually eating the cookies. And this is where I got the sticky fingers from, Vincent, is that the genes for BTEX-N degradation were all present in the genetic soup that they pulled out. And Michele, this is the one piece of the story that I'm most interested in hearing. How did they think to do the next experiment, the active viral predation in the deep subsurface? And they did this in a very clever way. Rather than looking for phage, they asked the question, in the microbial genomes that they were sequencing, were they able to detect the CRISPR-Cas system? And CRISPR, recall, stands for clustered, regularly interspaced, short, palindromic, repeated sequences, which is CRISPR associated. And these sequences, recall, act as an acquire immunity to viruses. And so the CRISPR-Cas frequency estimates range from 81% for the archaea and 40% of the bacterial genomes in cultivated microbes to 10% of archaea and bacterial genomes in the metagenomic data sets. And in contrast, 100% of the 3 archaeal and 84% of the 31 bacterial genome bins that they pulled out of the Marcellus samples had evidence of CRISPR-Cas system, with type 1 being the most prevalent. And so this is indicating that, or suggested to them, that what's actually going on is the bacteria are being attacked by viruses, and they're surmising that they're releasing materials into the community to effectively provide nutrients and provide osmoprotectants, and it's doing all sorts of remarkable things.
The ecology is just fascinating, isn't it?
I mean, this is just but step one. Go ahead, Elio.
Now, let me add a little something. This study is going to have to be done in many, many other settings. Let me tell you why I say that. I just pulled up a site on the internet called FrackFocus, which is the Chemical Disclosure Registry, and it lists what chemicals are used in fracking, and the list is humongous.
Oh, yeah.
I'm eyeballing this to be at least 50 different kinds of compounds. So this particular study is with a certain set of chemicals which they use. Other folks use different ones, and therefore the study has to be done in that context as well. So it's going to be quite complicated.
It is one of the reasons they did the study in 2 different sites, to ask if they saw parallel ecologies developing. But your point's well taken.
And that's what they did.
You have to take into account what chemicals you add. The function of the products, they listed here by classes, is to be a biocide, kill the bacteria, a breaker, a clay stabilizer, corrosion inhibitor, cross-linker, etc. Just goes on. So there is about a couple of dozen reasons why you want to use certain chemicals, and for each reason you have a collection of different ones. So I have a feeling this is going to be quite extensive.
But the environment in each of those is going to be hot, anaerobic, high pressure, and high salt. So those are the selective pressures that will be operating in every site.
But you don't think it's going to make a difference what it is that you pump in?
Pardon?
Don't you think it's going to make a difference what chemicals you pump in?
Oh, absolutely. And the product that you're trying to extract.
That's my point.
And that's effectively what this last 2 sections of the paper are. They're asking your question, how reproducible is this? And that was the point of the Utica shale, and as well as the point of asking the question, how important this GB fermentation product is. And when they amended the Utica shales with GB, they found a very different result than they saw in the Marcellus shale. And it was principally because of a different behavior that was actually going on. And they go on at great lengths to talk, and this is the subtlety that I think is best left to the readers of the manuscript to go over, because it really, you can get lost in the details if you don't have the paper in front of you to figure out what's actually going on here. But the take-home message is it's an elegant and very systems-dependent structure, and it depends on the input chemicals and the microbes that were associated with that input chemical structure, chemical matrix, as well as the hydrocarbon that they're trying to extract. And in the case of the Marcellus shale, they were going after gas, natural gas rather than oil. And you use other things for other things. And it depends on the amount of sulfur that's associated with the material, because you will use the thiosulfate as an electron acceptor, making sulfide, which can then poison the well. And so their conclusion that they came to is they were able to resolve the genomes from the Marcellus and Utica produced fluids, and they revealed, very remarkable microbial metabolism, adaptations, and most importantly is this notion that viral predation resistant mechanisms were evident in the fractured shales as a consequence of the viruses that are likely being introduced, the cutting materials, or the viruses are just sitting there waiting for an opportunity. And by lysing the bacteria, releasing more nutrients to the system, you can also begin to do other things. And from their 16S gene analysis, they could not have predicted this role of the halo anaerobium strains playing in fermenting this osmoprotectant, the glycine betaine, and the hydraulic fracking chemical additives such as ethylene glycol. And nor would they have been able to predict that the halomoniaceae with detrimental sulfide production. And it's just a remarkable paper. And again, that's why I caution people to look at the fourth figure first before you dive into the paper, because if you keep that figure in your head as you're reading it, it will become most relevant to you as you go through this. And
because the fourth figure is a cartoon image that lays out the logic of the study. It's a very nice visual abstract.
Very effective.
So Michael, is this bad? What's going on?
No, I think
is this bad news?
I think from a microbiological perspective, it's nothing that we didn't already anticipate would happen. If you put nutrients in, the microbes are going to grow. We've known this.
Is going to affect the ecological consequences of fracking? I mean, that's the ultimate question.
I think the answer is it may go after your groundwater, and you have to really begin to understand groundwater flow and all sorts of other things.
So and this is not good news.
We've given an infection to Mother Earth, and whether or not that infection is going to be detrimental or she can self-heal. But I think what this initial data are providing us is the resilience of microbial life to adapt, colonize, and habitat and invade a structure by physical and chemical features very different from their origin. And so the microbes are invading and they're adapting. Whether or not we'll be able to adapt accordingly. I think the thing that this will ultimately impact is in places like Pennsylvania, whether or not they do indeed have their groundwater protected and whether or not it's going to sour their groundwater.
Yeah, I think basically we have to keep an eye on this, right?
Yeah.
Yeah, and our listeners will be reassured to know that there is a lot of research going on in this area. So I was able to talk with Rebecca Daly, and she directed me to what's called the MSEAL project, MSEEL.org. You can read about it. It's work funded by the Department of Energy and has investigators from Ohio State and West Virginia. And they have two mines that are dedicated to research. So a lot of the other active mines are very difficult for researchers to get in there and do these longitudinal studies. But this MySeal project is designed to do exactly that. So it's an amazing team of scientists, geologists, chemists, and also people studying the social impact of the fracking in a particular area. So I encourage you to take a look at that website. And it's good to know that the Department of Energy is funding this work so that we can better understand the impact of these really engineered ecosystems that we are creating deep down in the earth. Microbes are going to grow, and we don't know what's going to happen. So it's great that they're taking such sophisticated technologies to watch what's happening over time.
And that's why I introduced this story by first talking about the earthquake, which is something that we've already appreciated, at least in the state of Oklahoma, by the increased frequency of earthquakes. If I said to earthquake to the general public, what state would you predict it would be in? Most folks would say California. Very few would say Oklahoma.
Right.
And so the issue is whether or not
there's no free lunch, is there?
Nope.
No free lunch.
Ever.
That's the take home.
Michele, did you learn something from Rebecca?
I did. I was able to talk with Rebecca last week. She is a research associate in Kelly Wrighton's lab. She went after she graduated from high school, wasn't exactly sure what she wanted to do. She didn't know if she was ready for college. She moved to Colorado Springs, took some classes there for a couple of years, but she was kind of bored by the introductory coursework. So she left school, stayed in the area, and got a job in a bookstore, and she stayed there for several years. She got a little restless. So after a decade of being in the real world, she applied for and earned a scholarship to attend Colorado College, which is there in Colorado Springs. So she earned a bachelor's degree there and then headed to Berkeley for graduate school. And that's where she met Kelly Wrighton, who is the senior author on this paper. Kelly and Rebecca were both graduate students at Berkeley. Kelly Wrighton was training with John Coates, while Rebecca studied subsurface chemistry in uranium mines with Mary Firestone, and she had a co-mentor, Owen Brody, of the Lawrence Berkeley National Lab. And so during that time, Rebecca contributed to 4 publications, but again, she was getting a bit restless. She got a call at that point from Kelly, who by this time had just been recruited to Ohio State and was looking to recruit a senior scientist. And so she recruited Rebecca as a professional staff scientist. So she manages the lab, she mentors graduate students, and has the luxury of leading her own research project. And thanks to the respect she had earned as a graduate student and a scientist at Berkeley, she was able to land this position despite not having finished her PhD. So it's there where she's been working for the last 3 years. And as you can see, she's making really interesting and important contributions. I'll add that her story kind of resonated with me because I know of other colleagues who have taken kind of an unconventional path to graduate school. Denise Monak is one person who comes to mind. She's now a full professor at Stanford, but she worked for many years with a bachelor's degree in Stanley Falco's lab and contributed to a lot of really important work in salmonella before deciding to go to graduate school and then launch her own independent career. And I'll say I maybe my story is not quite as extreme, but myself, I don't think I took the traditional pathway either. As an undergrad, I was never in a research lab except to track down a TA to ask him a question about class. I then worked as a lab technician for a couple years, and then my boss urged me to go to graduate school. I did that, but gave birth to both my kids while I was a student, and then took a year off to be with them before starting my postdoc. So I think there's a couple messages here, and one is that don't feel like you have to march right from high school into your professional career. There's a lot of opportunities to grow and get a variety of experiences, figure out what really excites you. And Rebecca shared that microbiology is the one thing she's done in her life that she's never bored. There's always something really interesting to do.
Nice.
And this particular project, she wanted to give a shout out to Joe Kretzky, who was the glycine beta-aine expert with his student Duncan Coons, and also this collaborative project that funded the work. So there is let me look at the formal name. They are funded by kind of like a program project by the NSF called Dimensions in Biodiversity. So this brings together multiple PIs to work on a common problem. So that's Paula Moser is the PI of that larger project. So between that and this MSEAL project, they're doing a lot of collaborative science with people with very different backgrounds. So I can see why Rebecca is just thrilled to be a professional scientist in the Wrighton lab. And I'll add that I happened to meet both Kelly and Rebecca when I was a guest of their student seminar years ago at Berkeley. So it's kind of neat to connect with them both again.
Nice.
Yeah.
Michele, do these environmental microbiologists, are they part of ASM? Is there a whole contingent of them there that are well represented?
At the ASM microbe meeting?
Yeah.
Are you asking?
Yeah.
I would not be going to those sessions, so I couldn't give you data on that. But if they're not,
there's certainly
we need
sessions.
to recruit them.
There's certainly sessions, and they're coming to the meeting, right?
Absolutely. Yeah, I remember hearing some very cool talks on the ecology in the ocean and how the rising temperatures across the world are affecting that microbiology and its long-term impacts. So
nice. Thank you, Michele.
Yeah.
That's a really cool paper. Thank you, Michael. And
you're most welcome.
Before wrapping up this episode, I just want to tell you about the 2017 ASM Scientific Writing and Publishing online course. This will explore the scientific writing and publishing process with ASM Journal's editors. It's an interactive webinar series. You'll hear about titles, abstracts, figures, figure legends, and the whole manuscript review process. This will take place from January through April of 2017. There is a registration deadline of December 1st. To find out more, go to bit.ly/SWPOC17. That's bit.ly/SWPOC17. You can find TWiM at iTunes or at microbe.tv/TWiM. Consider becoming a patron of TWiM and all the Microbe TV family of science shows. You'll go over to microbe.tv/contribute to find out more. And of course, we love getting your questions and comments. Send them to TWiM at microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you.
9-0, you said?
9-0.
Amazing. Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure, as always.
Thank
you.
You're welcome. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent.
And I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for technical help. I also want to thank the sponsor of this episode, CuriosityStream. The music you hear on TWiM is composed and performed by Ronald Jenkins. Find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.